Okay, we're live, everybody. Okay, so I'd like to thank everybody for tuning in. I'm very honored and excited to be able to moderate the first event of Black and Immuno Week. First, we have Dr. Keke Fairfax giving us our keynote for this week. And just to stay on track, I'd like to thank you all for joining us for Black and Immuno Week, a week where we will celebrate, amplify, and support Black voices in immunology. Before we begin, we'd like to thank our major sponsors, the Immune Deficiency Foundation and Imperial College London. Don't forget to tweet about our opening keynote using hashtag Black and Immuno Keynote, capitalizing the beginning of each word for accessibility purposes. Our presenters will be using captions with their PowerPoint presentations for our live audience. You can submit questions to Slido, which is pinned to our YouTube live, and or submit questions on Twitter using our hashtag. Again, it's Black in Immuno, capitalizing the first letter of each word for accessibility purposes. The videos will be captioned and posted on our YouTube channel, and transcripts will be available within 24 hours of the event. On our website, blackandimmuno.org. That's blackandimmuno.org. We also follow us on our social media pages, which are all at Black and Immuno, which is on Twitter, Instagram, LinkedIn, Facebook, and TikTok. And so, without further ado, we would like to welcome Dr. Keke Fairfax as our opening keynote speaker. For a brief bio, Dr. Keke Fairfax received her Bachelor of Arts from the University of Chicago in 2003 and her PhD from Yale in microbial pathogenesis in 2009. Her dissertation work focused on identifying novel fatty acid binding proteins in the human hookworm Aclystoma selanicum. She completed her postdoctoral training in Schistosoma mansoni and immunoparasitology with Edward Pierce and Gwendolyn Randolph in 2014. Dr. Fairfax began her independent laboratory at Purdue University in 2014 and then moved to University of Utah in 2018. At the University of Utah, the Fairfax lab has broadly focuses on using Helminth parasite Schistosoma mansoni as a tool to understand both the relative contributions of schistosome antigen versus IL-4 in inducing host immunomodulation and the complex interplay between lymphoid and stroma cells necessary to develop an optimal T and B cell memory response. And under this umbrella, she currently has 3 main projects. The first of which is understanding the immunological implications of maternal schistosomiasis. The second is dissecting the role of IL-4 in shaping the cellular environment of peripheral lymph nodes during homeostasis and antigen challenge. And 3, delineating the mechanistic role of antigen-driven immunological reprogramming and Helminth-induced protection from metabolic diseases. So thank you, Dr. Fairfax, for being with us today, and we really look forward to your talk.
Great. Thank you, Yvonne. So today is going to be less science and more my background and journey to how I got where I am. And then importantly, how I weave advocacy into my scientific and academic career. So I grew up in Detroit, Michigan. And I'm a product of the Detroit public school systems. I went to an absolutely fabulous school called Bates Academy, which was a school for the gifted that was a public school. And it had students from all across the Detroit area that attended. And there is really what really enabled me to be successful in my career. So it was a majority Black school, just like Detroit is a majority Black city and still is today. And I was raised within Black excellence. So everything I saw around me was high achieving Black fellow students, fantastic Black teachers. And, you know, we learned real, true Black history and the contributions that Black and brown people have made to the United States and world history in general. So I was always very firmly rooted and secure in my sense of self and my sense of intelligence, which is something that I know a lot of Black people in the United States do not have. And that's part of the pipeline problem that we have here. And so I was encouraged from very early on to pursue my deep interest in science. You know, in my early childhood, I was torn between wanting to be a paleontologist, and Jane Goodall was my hero. So I was also very interested in primatology. But I started doing, you know, truly professional university level science in high school, where my chemistry teacher, Pat Vance, you know, encouraged me and wrote me recommendation letters to apply for a program called HSHSP, which was run out of Michigan State University, that took high school students from all over the country and put them in labs at Michigan State for the summer. It was a 10-week program. And back then, I mean, that's fairly common today, but back then there weren't that many programs that did that in the 90s. So that was really critical for me knowing that I wanted to pursue a PhD. And I was in that program for 2 summers, the summer after my sophomore year and the summer after my junior year. You know, and I won science fair and also competed in the Junior Science and Humanities Symposium, won the state level, competed at the national level and the international level. And so that really set me up to be able to be successful really early in my undergraduate career. And as Yvonne said, I went to the University of Chicago, which was a great experience for me in some ways, and in other ways, you know, was really difficult. And so from an intellectual standpoint, the U of C was the best place for me. And I really loved the intellectual stimulation and environment. But there was no representation of successful Black STEM faculty at U of C when I was there. And, you know, so that was probably really when I started questioning, you know, the place for people that looked like me in science in America, was first in undergrad. But I had a fantastic undergrad mentor, Jean Greenberg, and I did a postbac with her before I went on to Yale for my PhD. And then, you know, when I started off at Yale, again, there were a few of us Black students. I had a really nice cohort. But again, there wasn't really representation of us as faculty members in the STEM. Of course, there is in humanities. And so the first Black female faculty member I met, you know, who had a STEM lab was my second year at Yale when I started working with the STARS program, which targeted freshmen who had been admitted to Yale with, you know, a less rigorous STEM background, who were URM, and wanted to catch them up and really give them the chance to succeed in STEM. So we taught them classes. They were in a lab, and they had a lab mentor, plus myself and a few other graduate students were their additional mentors to really help them get to be able to write and read science. And that was started by a fabulous Black female scientist, Dr. Black, who was a chemist. And that was really my first interaction with a Black female STEM professor. And that really solidified for me the need to make inclusion and increasing representation of URM in science a key part of my path in academia. I knew I wanted to stay in academia, but you know, it solidified for me that my career wasn't going to be just about doing great science, which I think I do great science, but it was also going to be about enabling more people who looked like me to be able to succeed. And so that's really been, a twin focus of the last 10 years of my career. And so I continued throughout my postdoctoral training to volunteer in programs that targeted URMs at the high school and undergraduate level, along with private tutoring, and really made that side by side equally important to me as doing my science. So, when I started my lab at Purdue, I didn't really have the opportunity to be my authentic self and do the advocacy I really wanted to do. I was not supported in that. And so when I decided I wanted to move my lab, I did so with the decision that I wanted to live an authentic life and be the role model I had wished I had my entire scientific training. That's not to say I didn't have fantastic role models. I actually absolutely had amazing role models and mentors who still support me to this day. And I would not be where I am right now without them. But there's a lot of things that you can't be taught by somebody who doesn't walk the same walk that you do. And, it's not easy to navigate academia and teaching, particularly if you're teaching in a predominantly white institution, as a Black faculty member, male or female. There are issues that have to be contended with that, my white colleagues just don't have to deal with. And so having a role model that can really help you through that, I think is what's key for being able to retain minorities in STEM. And so that's a really big part of my career goals and career paths. I want my legacy to be equally about what I've discovered in immunoparasitology as it is about the people I've trained and enabled to have successful careers in science. And so that's actually sort of why I work on what I work on. And so, when I started graduate school, I worked on bacteria like a lot of other people. And I knew I loved pathogenesis, but I did bacterial pathogenesis. I worked on Pseudomonas syringae, which is a plant pathogen of Pseudomonas. And when I decided to go to Yale, I think everybody assumed that I would go to a lab that was a collaborator of my undergrad lab that worked on Pseudomonas aeruginosa. And I did do a rotation there. But, I liked it and it was great. And Barbara was a great mentor to a couple of friends of mine. But, it was clearly not my passion. And I went out on a limb and decided to rotate in a hookworm lab. And that ended up being the lab I joined. The PI was Mike Capello, who to this day is extremely supportive of me. But why I fell in love with immunoparasitology is really because parasites, particularly helminths, are extremely neglected, but on a global scale, they have a far greater impact on the health as well as the economic opportunities of people that look like me. So this will advance, there we go. So I did my PhD, as I said, in hookworm infection. And there are 3 species that infect humans, Ancylostoma duodenale, Solanacum, and Necator americanus. Prevalence is really high in endemic areas. Like all helminths that infect humans, there's no acquired sterile immunity. Reinfection occurs quickly following antihelminthic treatment. And heavy hookworm infection is associated with anemia and malnutrition and high disability adjusted life years. And now this is endemic in sub-Saharan Africa, South and Central America, and really extracts a heavy toll on particularly African children. And so that's really what drew me into immunoparasitology, was the chance to make a difference to hundreds of millions of kids that looked like me. And worms are complicated, extremely complicated, far more complicated than bacteria or viruses. The co-evolutionary forces that have exerted themselves over hundreds of thousands of years as we've evolved together have allowed them to live for sometimes 50 years in a host. And that takes a long toll on the person that's infected, but also speaks to what a true pathogen is, which is the ability to coexist with a host for a long period of time. And so that complexity has always been really intriguing to me. And so I did a lot of work in hookworms on fatty acid binding proteins and their role in pathogenesis. But hookworms are complicated because they don't have a genetically tractable rodent model. There are a few non-human primate models, but are difficult for other reasons. So all of my work was done in the hamster model. And we just don't have really great immunological tools in hamsters. So when I was looking to do my postdoc, I knew I wanted to become a far better immunologist, and I wanted to work in a model that would enable me to do that. So I decided to switch to schistosomiasis. And I started working with Ed Pierce. I started working with Ed Pierce, and I started working on Schistosome-driven. And in the 5 years of my postdoc, I became a 100-fold better immunologist than I was before I started the postdoc. And it cemented that I would have lifelong work in schistosomiasis. So schistosomiasis is endemic in sub-Saharan Africa, South America, and the Caribbean, as well as Southeast Asia, particularly China and the Philippines. And I work on Schistosoma mansoni. The vast majority of my work is on Schistosoma mansoni, which is really a high disease burden in sub-Saharan Africa and in Brazil, with a little bit in some other South American and Caribbean countries. But this is really a disease of sub-Saharan Africa in terms of pathology for Schistosoma mansoni. And so this is a chronic infection. Like with hookworm, there is no sterile immunity. So kids are infected really early. Most children are infected before the age of 5. And even with deworming campaigns, children are generally reinfected within 3 months. And the effect is lifelong liver inflammation, fibrosis, and damage. And so the disease is actually not due to the adult worms. It's due to granulomatous inflammation around parasite eggs. And so these are granulomas in the liver. The end result is hepatic fibrosis and, in severe cases, portal and pulmonary hypertension. And this is really thought of as a type 2 disease. So there's a marked Th2 response with eosinophilia, alternative activation of macrophages, and the production of IgG1 and E in murine models, IgG1, IgG4, and IgE in humans. And so these worm pairs can live up to 50 years in a host and cause chronic disease as well as chronic immunological problems to other pathogens and immunizations. And so this is really my lifelong focus. And I couple this with my advocacy work because I while I'm a product of colonization, I really don't like colonization. So my point of view is that Western scientists should not just do experiments in sub-Saharan Africa. We should strive to educate and empower people in sub-Saharan Africa, as well as India and other countries, to have the knowledge and the training, and importantly, the advanced tools to help solve their own problems. And so I work on a disease of sub-Saharan Africa, but I also have a strong commitment to training scientists from LMICs. And so I'm going to talk a little bit about the mouse work that we do, but then I'll also tell you about how I'm putting this in place in the human pursuits that we're aiming for. And so, as Yvonne said, my lab has a pretty broad focus on type 2 immunity and homeostasis and disease. We have 2 chronic schistosomiasis models that we work on: maternal schistosomiasis, which I'll talk to you a little bit about, and then Helminth modulation of metabolic disorders. And then we do have a basic immunology side in the lab that is really centered around IL-4. And you know, I hope my work over the next 10 years will really reposition IL-4 as not just a type 2 cytokine, but as a homeostatic cytokine. And so we look at the cell intrinsic versus cell extrinsic functions of IL-4 in the stromal lymphocyte axis, and the role of IL-4 in homeostasis of hematopoietic progenitors. And so give you a little bit of background about maternal schistosomiasis. Maternal parasitic infections in general are an extreme problem in sub-Saharan Africa and South America. And we first started to realize this with the finding that relative vaccine efficacy in lower middle income countries is often significantly lower than is what is realized in the developed world. This is likely multifactorial, but there's strong evidence that the chronic presence of prenatal and early childhood parasitic infections does undermine the development of effective immune responses to specific vaccines. So in the case of schistosomiasis, there are 40 million women of childbearing age in the developing world that are at serious risk for schistosomiasis, and at least 10 million women in Africa are diagnosed with schistosomiasis in their childbearing age. And so we really wanted to couple the power of mouse models to do mechanistic work to hopefully provide a lasting impact that's going to improve vaccine efficacy in lower middle income countries where schistosomiasis is endemic. And so we have, I'm not going to go into a lot of really complicated data because this is a really broad audience, but we use tools like transgenic reporter mice, as well as confocal and multiphoton imaging to mechanistically dissect the B and T cell and stromal cell interactions in offspring born to uninfected mothers versus infected mothers, in order to figure out mechanistically how in utero exposure to egg antigens might be modulating offspring immunity for a long period of time. And so this is a lymph node from an uninfected mother, an offspring born to an uninfected mother, and this was vaccinated right at weaning. And it's vaccinated with a commercial human tetanus diphtheria vaccine. Complement receptor 1 and 2, CD2135, are in red. GL7 marks the germinal center, and blue marks FDCM2, which are follicular dendritic cells, which is the stromal cell that is really critical in the B cell follicle for providing help for class switching as well as memory B cell survival. And so offspring born to uninfected mothers have a lot of germinal centers in a draining lymph node. And if you look at an individual germinal center, you see that there's a nice large germinal center. There's a cap of CD2135 FDCM2 double positive cells that are the follicular dendritic cells that cap the B cell follicle. And they present immune complexes to the germinal center B cells. In offspring born to infected mothers, there's fewer germinal centers. They're all smaller and less bright, and they don't have a cap of follicular dendritic cells. So they're not physically able to have this cell-to-cell contact with germinal center B cells that exists in the offspring born to uninfected mothers. We believe this is the base mechanism for impaired immunity in these models. We've used other tools. This is single cell RNA-seq on that same germinal center reaction. And I'm only showing you follicular B cell and germinal B cell clusters. But we discovered using offspring born to 3 different types of mothers: uninfected mothers, patently infected mothers that have egg antigen exposure, and single sex infected mothers that are only infected with either male adult worms or female adult worms, so there's no eggs being made. That in utero exposure to egg antigen only drives transcriptional modulation in all of the follicular B cells as well as in the germinal center B cells. And so there's a reduction of a critical transcription factor for B cell identity, EBF1, as well as increases in Jun and Jun B, which would decrease the cell's life, and decreases in RBX1 and TMSB10, which are important for cell cycle and B cell receptor signaling here. So there's a defect in both B cell identity and proliferation, as well as B cell receptor signaling, which would be absolutely critical for developing a memory B cell response to immunization. And so, you know, we validated the single cell data via flow cytometry. I won't get into the details, but we do see that in plasma cells, which are the circulating effector cells that would secrete antibody, there is a reduction again in this B cell identity transcription factor, as well as in a reduced ability to proliferate, as marked by Ki67, and that's at steady state. And then also that is true at the peak of the B cell response following immunization. The germinal center B cells and the plasma cells have reduced EBF1 transcript and reduced cell cycle with Ki67 proliferation. And so this work will be published soon. It's the minor revision is just going back in at PLOS Pathogens. But this has really set us up to have outcome measures we can now use and translate into humans for a new human maternal schistosomiasis study to really see, is this the same defect that is happening in humans? And then can we intervene and rescue this defect in B cell identity and proliferation? And so that is, again, where my advocacy desire for training overlaps with my actual science. And so I have always wanted to only do human studies in an African country, if I could do so in conjunction with scientists from that country that would involve training the next generation of scientists from that country. And so I've spent 4 years now trying to come up with the right situation where we can enable success in this venture. And it was really moving to Utah that has enabled this to happen. I have a fantastic colleague here who's a pediatrician who is Ghanaian and has always wanted to be able to go back and do meaningful clinical research in Ghana. And, you know, my career has sort of come full circle, and I've been connected back with a scientist at the Noguchi Institute in Ghana, who I actually knew since I was in graduate school. He has a very long and strong collaboration with my PhD advisor. And so we are setting up to begin a pilot study of these transcription factors, as well as some other cellular outcome measures that I didn't get into here in Ghana. And when that is submitted for funding as an R01, it will contain funding for training 2 Ghanaian scientists. And so I really want 20 years from now, there not to be a need for American scientists to put all of the effort into getting funding for research that's going to benefit communities in places like Ghana and Kenya or even India. I would like Ghanaian and Kenyan scientists to be well enough trained that they can compete with European and American scientists for grant funding that will enable them to really chart their own destiny in terms of providing meaningful scientific advances for the problems that affect their communities. And so I have a really deep commitment to that training portion. And also, of course, to serving as a mentor and role model for Black American students and Hispanic students as well. And then I'm not going to show any data on this because we are already over time. But the second project, is also really rooted in schistosomiasis. But here I'm hoping that the end goal deliverables will help people that are infected with schistosomiasis, but also provide a benefit to Western society and have new treatments for cardiovascular disease and diabetes and the complications that occur from that. And this project stems from an observation that we've known for 20 years, which is that globally helminth infections inversely correlate with metabolic diseases. They also inversely correlate with allergic and autoimmune diseases as well. But this project focuses on metabolic diseases. And the pretty shocking statistic that in the Western world, so in Europe and America, more than 65% of diabetic patients actually die from cardiovascular complications. And insulin resistance and obesity are the 2 most important risk factors for the development of coronary artery disease in diabetics. So of course, there's a critical need to develop innovative approaches to reducing the pathology of metabolic diseases. And this project is really focused on taking advantage of the fact that helminths are very immunomodulatory because they're a co-evolved pathogen in order to identify novel proteins that we can use for good. I don't advocate infecting people with any helminth infection, although those trials have been done. But the desire is to find helminth antigens that are going to give all of the beneficial effects of helminth infection, in this case on the metabolic side, without any of the pathology. And we've made some real advances in that. And I just today resubmitted that paper to PLOS Pathogens on the macrophage mechanism that we have uncovered. So I will just jump to, and I can't, sometimes I can't technology here. The beautiful faces of the people that I have mentored over the last 6 or 7 years.
Thank you, Dr. Fairfax. That was amazing. I got a text from a friend saying that was very inspiring. So
sorry, it's always, I don't do that many not pure data talks. So I'm always a little more nervous about those, but.
Yeah, I think we got really good reception and we have a lot of questions. So I'll just start with the first one from the Slido. This is from Tanya Aaron, not to assume their pronouns. They ask, how were you able to determine which institution would support you in science and advocacy during your job search? And how did you advertise your skills in both?
Okay, so this is actually really critical. So when I made the choice that I was going to leave Purdue, and it was just a question of where I was going to go, I made it very clear in all the places that I talked to, what it was I was looking for from a boss, so from a department head, as well as, what I wanted the support to accomplish. So I have two absolutely fabulous bosses here at Utah. So I have a department head, Peter Jensen, who is also the CEO of ARUP. And then I have a division chief. Because we're a clinical department, we have a lot of divisions. And so each division has a chief. And my division chief is Brian Evavold. And so in my first interview with Peter, I made it clear to him that I can be a troublemaker, but a good kind of troublemaker. And that I was looking to make institutional level changes that were going to have a lasting effect on representation in science. And Peter was 100% here for it. He already had a goal that he wanted to improve the diversity of the Department of Pathology. And so, he was very happy to find in me somebody who was willing to tell him what might work and really push for those things to happen. So I took the job because I believed that Peter and Brian would always have my back, and that has been shown to be true over the last 2 years. And, Peter has put a lot of trust in me and given me the title and the tools financially and infrastructure wise to really try to make changes. So I'm currently setting up a summer research program that is going to focus, at least for the first year, on Black students, Black undergraduate students, but I would like to expand it in a year or two to also partner with majority Hispanic serving institutions. But we right now are developing partnerships with 2 HBCUs. And we will have at least 8 students on campus this summer for a summer internship program that is joint, that I've set up to be joint with the Department of Pathology and the Diabetes and Metabolism Research Center, which I'm also a member of, which spans quite a few departments. And so from a financial standpoint, I have the support to do that. From an infrastructure standpoint, I've been given the support for these initiatives. I'm the director of diversity, equity, inclusion, but I also have a committee with 2 other fabulous women who are also committed to institutional level change. And both of them are more in the clinical side of our department. And so we're putting together plans for increasing URM recruitment to our fellowships as well as our residency program. And then I'm actively involved in increasing the diversity of our faculty through both broad faculty searches, which I was on the faculty search committee this past year, as well as targeted recruitment efforts to a more limited set of faculty candidates that will allow us to increase both our ethnic diversity as well as the number of women in the department. And so I'm really supported in that. And I took the job because I believed Peter, and he's shown himself to be a man of his word and extremely supportive of me no matter how much trouble I cause.
That's awesome to hear. I actually graduated from HBCU undergrad, Hampton University in Virginia, so it was really good to hear. We have a lot of questions. We probably won't get to all of them, but I will just go in order to be fair. So the next question is from Elaine. Would you say that the parasitic worm impairs the education of B cells, or do you see changes in B cell repertoire in infected compared to uninfected mice?
So it's actually far more complicated than that, and it is not in the paper that we're putting the minor revisions back in. It'll be the next paper, so I don't want to give away too much from my current grad student, Lisa, here, and she's here in the Jurassic Park shirt. So while there is an impairment of peripheral B cells, we have a significant amount of data that indicates it happens at the progenitor level. And so we are currently in the process of analyzing the sequencing of progenitors. We have a couple of long non-coding and microRNA targets that we believe are regulating chromosome accessibility at the progenitor level. And Lisa actually on Monday is doing some really critical fish experiments on that side. And so that story we're hoping will be ready to be submitted by February or March. But it's actually far more basal of a mechanism than impairment of peripheral B cells. So the most striking thing that will be in that paper, but I didn't show you, is that those follicular B cell changes actually happen at every single developmental stage of the B cell in the lymph node, which is what first led us to the progenitors. It's a pretty universal defect. There is also, I didn't get to it at all here, but it is in the paper, there's a defect in the stromal cells, in the follicular dendritic cells themselves, that we believe amplifies this issue. We're just now, I did the cell transfers 2 weeks ago. I think Lisa should be analyzing the data probably on Monday or Tuesday. But we've started to do the experiments to figure out how much of the defect is coming from the stromal compartment versus the B cells. So we've done some cell transfer experiments to address that question. We do believe both sides of the axis are defective, and that the FDCs can't properly instruct the B cells, but that also the B cells are impaired from the time that they come out of the bone marrow. And that's all in the mouse model. And these are so we have outcome measures that we can test peripherally in humans. And the IRB for Ghana has been submitted. We're hoping it'll be approved by January, and that we can start recruiting pregnant women for our dyads by February to get preliminary data to see if these B cell defects also are occurring in humans.
Thank you
it's
so much.
harder to test the stromal cells because, we're not going to take out kids' lymph nodes.
Thanks for that answer. The next question is from Donald Palmer. Could you further discuss the difference between infected and uninfected mothers? How much is this related to programming of the fetus immune system?
So we know that it is dependent on a certain level of antigen exposure in the mothers, and we measure that by maternal titer against schistosome egg antigens. As I said before, it is egg antigen dependent. And so there is a threshold, there's a lower level to which if the mother's titer is below that, the offspring aren't reliably modulated. As long as she's above that, they are modulated. And, there isn't like a 10-fold increase as you go up in log titer of the mother. There really just seems to be a threshold. And it's not dependent on the infection stage of the mother. This came up, one of the reviewers asked this. And so it's taken us a long time to be this good at this, but we actually now can have mice have their first litter right during acute infection. So they get pregnant at about 6.5 weeks, right when egg laying starts. And we're able to get, from really good mothers, we're able to get 5 litters or so. We're able to get litters through 26, 30 weeks of infection. And there is no difference. As long as that first litter, the mother is at above that titer threshold of where we see modulation, every litter she has after that will have the same degree of immunosuppression in the offspring. So it's not just acute disease versus chronic disease. It's really related to the maternal egg antigen titers which for me, having worked in schistosomiasis for this long, the titer level is really a measure of the amount of egg antigen that's circulating, and how well the immune system responds to that. We're still working on experiments to suss out how much free antigen is transferred across the placenta versus whether or not this is maternal immune complexes transferring or actually maternal immune cells. We do have some preliminary data that suggests some maternal memory B cells are transferred. It's not quite ready for publication yet. We want to do it one other way to be 100% sure. But we do believe that maternal B cells at least are transferred to the offspring. And so that can come, those can come bearing antigen. And so we're not quite sure how much of it is antigen actually in the offspring itself, modulating these progenitors in the bone marrow, or maternal B cells trafficking to the offspring and doing the modulation. So we're still working on mechanistically which level that is. At the end of the day, I think the answer will probably be that both things happen. But that's the level we're still trying to suss out the detail on.
Thank you for your answer. Okay, for the next question is from Jay Ortiz, who's actually my lab mate from UPenn. He asks, has your group also looked at whether there's also dysregulation of T follicular cell responses that accompanies this defect in B cell cycle regulation?
Yes. So an individual TFH cell is not impaired. Where the impairment occurs is in differentiating into TFH cells in the first place. So you have your reduced Th2 cells and you have reduced TFH cells and reduced memory CD4s. And those are also impaired across all developmental stages of the T cell. And we also have data that they're impaired at the progenitor level.
Cool. We have another question from Elaine. I think it's an important question, so I'll ask it. How do you maintain the balance between advocacy and being a full-time PI? And what advice would you give to grad students or of color, especially Black students?
So, I mean, it's definitely not easy. I will tell you, Lisa all the time tells me I do too much. And I love my lab. I keep it real with them. They keep it real with me, you know. And so Lisa's told me I need a vacation. It's not easy. And I would say that I don't think I would be successful if I had been this hard on the advocacy side early on. I think, you know, you have to get your lab established and your science established, and then you can start to deviate some of your attention. And so, you know, I was not this vocal of an advocate when I started as an assistant professor. And I wouldn't recommend that anybody do that. When I got more comfortable and my first PhD student was getting ready to graduate, and, you know, I had recruited Lisa at that point. So Lisa was actually an undergraduate with me originally. And then she was my tech for a while. And then she moved to Utah with me. And then I actually talked her out of going to vet school and instead to doing a PhD. And then she joined my lab as a PhD student. So then I was sort of set from a continuity standpoint. I still do a lot of hands-on work in the lab. As I said, for better or worse, I still do the vast majority of the cell transfers for the lab. I do a lot of the sorting for the lab. But the lab can do enough on its own that I can take more time to work on other things. And so I think you really have to be sort of set and on an okay trajectory before you start to deviate your attention, because there's no benefit to going hard on advocacy if you can't actually be promoted, because then you're not going to have a job, and that's a problem. So you have to make sure that it's not going to hurt your scientific side, and that you're comfortable enough in where that is in order to go on. And I will say it's still a tough balancing act. You know, there are days that 6 hours of my workday are spent on either administration or service stuff. I also do a lot of service for ASTMH. I'm a counselor for ACMIP, American Committee on Immunocellular and Molecular Parasitology. And there I do a lot of also trainee-focused things and advocacy things. And so I spend more time on service than a lot of faculty members do. And it's just, you know, I probably vacation less than other people. That's not a good thing. I don't recommend that to people. But, you know, you have to figure out what works for you. I do still spend a lot of time with my kids. I have 2 small children. And I still do hands-on lab work. But I care a lot about the advocacy side. So I'm willing to put off working on a manuscript or a grant until 9 o'clock at night after my kids go to bed so that I can do other tasks during the day that are administrative and service. It's really about your priorities. And I will say, as a woman with children, once you have kids, you learn to be far more efficient in your time and than you were before kids. And so you amazingly get the same amount of work done in less hours in the day.
That was a good anecdote. But this will probably be the last question. We do have an event at 2 p.m. of the networking. So the last question will be from Ada, sorry about if I mispronounced that. Weinstock, are the impairments in germinal centers and B cell responses rescued if pups are raised by an uninfected dam?
That is a very good question. So there is a lab in Brazil that has done cross-nursing. And they haven't published the same cellular outcome measures that we look at. And they haven't done any response to immunizations. But what they did look at was co-stimulatory molecules on B cells. And the most suppressive event is being born to an infected mother, not being nursed by an infected mother. And being nursed by an uninfected mother doesn't overcome the defect in co-stimulatory molecules in their model. And so to me, that would suggest that the most critical window is in utero. And that fits with all the data that we have based on where we think the modulation is happening during progenitor differentiation. That would be in utero events, not early postnatal events. But we will probably, in about 6 months, start doing some cross-nursing experiments in the context of our progenitor experiments to determine the contribution of nursing exposure. We do know that there are egg antigens in breast milk in both humans and mice. That's been published by a couple of really great groups. And so it's not a flippant question by any stretch of the imagination to think that breast milk would be modulatory. There are immune cells in breast milk, and when we know there's antigens in breast milk. But for the phenotype that we have found, we think the most important stage is in utero. But we still need to formally prove that. And we're gonna focus on doing it in the context of the progenitor stuff, not the immunization stuff.
Thank you, Dr. Fairfax, for an amazing talk, for amazing anecdotes throughout, inspiring us all here today. We are here for today. Sorry about the captions. I don't think they were working, but we will have transcripts out by tomorrow. Sorry for everyone whose questions we did not get to, but we will continue to collect the questions for Dr. Fairfax and to answer live through Twitter, again, using our hashtag Black and Immuno Keynote, capitalizing Black and Immuno, Keynote, the first letter of each of those words. And lastly, we do have our networking session at 2, which will begin shortly. Thank you for everyone who tuned in live. And this video will be available in the future as well.
Thank you.